Perfect.
Yes, sir.
Yes,
Okay. Okay, thank you. So let us start the class. So the subject is medical virology and mycology, and the subject code is BMLS 4005. And currently we are in the unit one. In the unit one, we have introduction to medical virology, introduction to medical important viruses, structure and classification of viruses, multiplication of viruses, collection, transport, and storage of viral samples for viral diagnosis. So these are the topics. And so far we discussed about introduction. We discussed about medical. We discussed the structure and classification of viruses. We even discussed about the multiplication of virus. And in today's lecture, I will discuss about the cultivation of viruses. So a quick overview. So we discussed the definition. Then we discussed about their size. Then we discussed about other parameters like their replication, then their other classification, then their families, their classification. We have DNA viruses, RNA viruses. Then we discussed about other content. Then we discussed their morphology in detail, like size, symmetry, shape, chemical properties, and resistance and susceptibility of viruses. Then we discussed about their replication in detail. In the replication, we also discussed about ligands and incubation period and eclipse phase. So eclipse phase means virus DNA will be embedded within the normal cellular DNA or genome, so that virus will be completely hidden from the immune system and from the blood. Such mode of viral phase is called as eclipse phase. Then we also discussed about incubation period. Incubation period means it is the time taken for the virus to attach to the cell surface and release of virus. The time taken for the attachment of virus till the release of virus is called as incubation period. And the incubation period will be varied from virus to virus. So this is what we discussed so far. Then the next topic will be viral cultivation. So today I will discuss in detail about viral cultivation, how to grow or how to cultivate a virus in the lab. Okay. Uh, so let us see. So for example, just like the way we are doing bacterial cultivation or bacterial growth medias, here we are going to grow the viruses. Rather than saying growing viruses, we will say cultivation of viruses. How we will cultivate the virus. So there are many approaches. So first of all, if you want to cultivate the virus, we need a living cell, right? So we need a living cell for the replication of the virus. So this living environment can be taken from three areas. One is we can use animals. We can induce virus directly into the animals, and we can cultivate the virus, or we can take embryonated egg for the cultivation of virus, or we can also take tissue cultures for the growth of viruses, for the replication of virus. So we have animal inoculation, embryonated egg culture inoculation, or we can also use tissue cultures. In the embryonated egg, we have different areas. There are different parts of the embryonated egg, like a cam membrane, allentoic membrane, amniotic sac, and yolk sac, that we will discuss later. And then third one is a tissue culture. In the tissue culture, this tissue culture has further divided into three types. We have organ cultures, explant cultures, and cell cultures. Organ cultures means here we will take entire organ. Say for example, if adenovirus grows in the adenoid tissue, then we will take a piece of adenoid organ, tissue organ, and we will grow the virus. If you want to grow any respiratory viruses, then we will take a piece of trachea and we will grow the virus. So we will take if it is hepatitis, then we will take a piece of liver and we will grow the virus. We will replicate the virus, or we will cultivate the virus. So this is called organ cultures. Then explant cultures. Explant cultures means from this organ, if you are taking a small bit, then that is called explant culture. Then we have one more culture called cell cultures. So here we will take only individual cells of the system, and those cells will be utilized for the replication of the virus. So these cells will be artificially grown in a cell media. 1 second, students, 1 second. All right. So this is the methods of cultivation of viruses. We have animal inoculation, embryonated egg, and tissue cultures. Then let us see first animal inoculation. So the idea is first we will take the patient sample, blood sample, and suspected with viral infection. So this blood sample will be injected into the animal. Now, if the patient has any respiratory virus like COVID virus, okay, coronavirus, then we will inject coronavirus directly into the rat tail or rat lungs. And if rat died due to some lung respiratory complications, then obviously that indicates the patient sample has a coronaviral infection. So we will inject this virus in multiple regions of the animal. We can inject virus in the tail, in the subcutaneous, or in the veins, in the intracranial, in the brain, or even in the individual organs of the body. So upon injecting virus into the animals, they will get infection actually. They will get the disease and they will die. If they get disease, that indicates positive for the sample, positive of presence of virus in the sample. If they didn't die, then that indicates negative test. Okay, there is no virus in the blood sample. Even though if the test is negative, but still these animals need to be harvested. These animals need to be killed. There is no other way. So there are many ethics involving to use animals in viral research actually. So this is the problems with animal inoculation for viral cultivation. So let us see few things. So inoculation of virus in animals. So lab animals play an important role in viral pathogenesis. So live animals, what animals we are using means we will use monkeys, mice, rabbits, guinea pigs, ferrets. Ferrets means they are a type of rats, very fast running rats. Okay. Um, Ferrets we are seen in some cartoon. Okay, and mostly mice is used in mice is used as a regular lab animal for inoculation of viruses. So I will show you few pictures of this one. They are really cruel actually. The way we are handling these animals is very cruel. So here we are using a monkey for the inoculation of virus, and this poor little monkey it is suffering because of the research purpose. We are really um, ending its life. Very painful to see. So yeah, so this is another rabbit used for viral inoculation, and rabbit got severe infection, skin infection by this viral virus. So this is how we will use animals as a candidates for viral test. What is your thoughts on animal usage, students? Whether we need to proceed with for virus research with animals? Is it wise to use animals for viral research? What is your thoughts on it? This is pakka animal cruelty, right? Are you listening?
Yes, yes,
Yeah,
yes.
yeah. This should not be the case. We should not show torture to the animals actually. So it should be prevented. It should be stopped. Is there any other way of doing viral cultivation other than animals? Okay. So, but there are a few advantages of animal inoculation. Okay, we can use embryonated egg or we can also use individual cells. But the problem is, let us see the advantages and disadvantages of animal inoculation. Can you tell me the disadvantages of animal inoculation? What are the disadvantages of using animals for viral research? What may happen? Tell me the disadvantages first. You just guess. Disadvantages are we are torturing the animals, we are showing cruelty towards the animals, we are killing the animals. Any other disadvantages?
But their immunity and our immunity have a
If
difference.
it's out
So
of
I
his
think
hand, then it
success is not
infects
successful.
virus a lot.
Mm-hmm.
So if
Mm.
But sometimes genetics material are matlab basically genetic material are different.
Mm.
our and animals, and immunity is also different. So it
Mm.
may be that some virus showing a positiveness in the
Okay.
animal and some are not showing in us.
Maybe they may give false results also. Just because virus shown infection in the animal doesn't mean the virus will show same infection in humans, right? That is correct. But at that at that cases we can change the animal. We can go with the very nearest human lineage. We can take primates, we can take gorillas, we can take chimpanzees, because chimpanzees and humans have very similar genetics. So obviously there is involvement of animals. We we are killing animals, and that is one advantage. What Abhishek said is exactly correct. Not all animal will just because animals show certain symptoms doesn't mean that same thing present in humans. So that is one disadvantage. Any others? Any other disadvantages of animal usage for viral replication? Any other disadvantages, students? What are other disadvantages of using animals for viral research? See, we are killing animals, we are showing cruelty towards the animals, and animals are not right models because we can't use humans there to test the viral things. Then other one is there is a chance of escape of these animals. We are working with we are doing a research on one particular virus, and we included this virus in a rat or rabbit, and there is a chance that this rabbit may escape from the lab, this rat may escape from the lab. If it is escaped, what will happen? Now it will spread, it will do contamination to the society, it will go to the wild, and it will contaminate the entire surroundings. It may become a pandemic also.
One more major
Hmm?
thing, sir. Sir, one
Hmm.
more major disadvantage is like leakage of the viruses, like coronavirus.
Exactly. Leakage of viruses can happen. Escape of virus happens because of these animals. Yeah, this may happen. So these are few disadvantages of using animals for viral research. Then, yes, I'm agreeing there are more disadvantages, but let us also see advantages of animal inoculation compared to other modes. Other two are usage of embryonated egg, just born embryo. We will use egg embryo for growth of virus, and we have individual cells also. We can take a single cell from the body, and we will grow the cell, and we can do viral replication or viral studies on the cells. What are the advantages of animal inoculation with other two? Can anyone guess advantages of animal inoculation? Okay, few advantages are, here we can able to understand the viral mechanism. Viral pathogenesis can be only understood by using live animals. Just a second, I'm getting repeated WhatsApp messages. Okay, fine. Um, see, uh, I cannot see pathogenesis, that means clinical symptoms, signs and symptoms in a embryonated egg. I cannot see signs and symptoms on a cells, but on a living animal, I can able to see complete stages, clinical stages of the virus, right? Immediately after infection, what signs and symptoms shown by the animal after and post infection. So I can able to study the pathogenesis of viral virus, and I can also study the immune interaction with the virus, how the virus is interacting with the immune system, what cells has been increased, what cells has been decreased, what cytokines are increasing. So uh, always studying animal model will give more data to us compared to other two. Animal models will give more data to us, which will be very useful for research purposes. So yeah, this is the primary advantage of animals. But uh, yes, I do agree the disadvantages. We need to sacrifice the animal. So that is why, unless and until it is really important, if there is no other way to understand the virus, only at that time we should use animals. There should be stringent policies and rules to use animals in the research. There should be a very strong ethical guidelines should be designed to protect the animals. So yeah, these are the advantages and disadvantages of animal usage in viral research. So let us see this animal inoculation. So animals are used for primary isolation of certain viruses to study pathogenesis of virus diseases, to study viral oncogenes. Some viruses can cause cancers, right? So those cancer causing viruses can be studied by animal inoculation. And for this purpose, we can use suckling mice, guinea pigs, rabbits. So all these animals can be used. So the virus will be inoculated in multiple methods. It virus can be inoculated into the cerebral, that means into the brain of the animal, or under the skin of the animal, into the peritoneal, in the abdomen of the animal, or even in the intranasal route of the animal. So by introducing virus in these routes, either in the brain, in the nose, in the abdomen, or in the skin, we will study the virus. The primary advantages of animal inoculation are for primary isolation of viruses, to study pathogenesis of viral diseases and to study viral cancers, oncogenes, we will go with the animal inoculation. And disadvantages are we need to kill the animal, we have to be cruel on the animal. There is a chance of escape of virus from the lab due to mishandling of animals, and it is also hazard for humans. The injured animal will show aggressiveness. Injured animal may bite us. So there are few disadvantages and there are advantages also. So by comparing this, in the beginning, scientists are using extensively animals only for viral studies. So millions of rats, millions of rabbits, millions of lakhs of monkeys has been sacrificed for the purpose of research in viral research. So these need to be stopped. There should be some alternative methods to study viral replication. And those alternative methods came by the name of a scientist called Robert Gallo. So he will be our second scientist. So Robert Gallo, his name is Good Pastor. His name is Good Pastor, and Good Pastor given a new idea that why can't we use an embryonated egg? We can take an egg and we can incubate it. So within 12 days, we will get an embryo. It will take 22 days to become a full grown chicken. So we can do our viral studies on this just unborn embryo. So he suggested the usage of embryonated egg, and this technique has literally um, safeguarded millions of animals actually. So after invention of embryonated egg culture, millions of animals has been prevented from sacrificing for viral research. So that is a really good move in medical research. 1 second, let me check whether it is recording or not. Yeah, this is recording. So now this is a good pastor. So animals loves him a lot because he saved them. So what good pastor said is, we can take a embryonated egg and we can introduce different types of viruses in different routes of the embryonated egg, different locations of the embryonated egg. So even I myself has worked with this embryonated egg for studying some oncogenes and viruses, viral related oncogenes. So here you can see this is an embryo and these are the blood vessels, and you can clearly see the beating heart of this embryo actually. So in this embryo, we are trying to isolate certain oncogenes and some viruses actually. We are doing some drug properties on this one. So as embryonated eggs will be used for studying the viruses. So let us see the structure of an embryonated egg. Now I will draw the structure of embryonated egg. So kindly pay attention. Very important. So now I will show you the structure of an embryonated egg. Let me share the screen again. So this is very important, students. Embryonated egg culture. Embryonated egg culture is very important as per your syllabus or everywhere for your lab practicals for anything. So to draw the embryonated egg, first you can do like this. First draw a cross, okay, a cross like this. Then from here, draw the outline. See, now I got a proper egg shape. Now I will erase the inner cross. So this is our egg, okay, eggshell. Now I will draw inside out. So first, this egg shell will have two outer membranes. So okay, I'm drawing two outer membranes. If you boil the egg, students, when you are boiling and if you are observing the egg, you can see one small transparent layer under the egg actually. You may seen it. Every one of you may seen a transparent layer like a onion layer on this egg. So this onion layer is
Yes,
called
sir.
as, yeah, this membrane is called as cam membrane. Cam membrane. CAM membrane. What is this cam stands for? Cam stands for chorionic allantoic membrane. CHORONIC. Chorionic allantoic membrane. CAM membrane stands for chorionic allantoic. Allan allantoic membrane. Chorioallantoic membrane. I'm not writing membrane here. Okay. So these two layers are chorioallantoic membrane. So this is a double layer membrane. Now in between these two layers there is an empty space. There is an empty space. This space is called as allantoic cavity. This empty space between the two cam membranes is called as allantoic cavity. Allantoic, Allantoic, allantoic cavity. Really tough to write with the touchpad. Allantoic cavity. Okay, so cavity. So in the center, in between the two layers, we have a cavity called allantoic cavity. Then on the top, students, I didn't shown here. Sorry. On the top, you have an empty space, students. On the top, you have some empty space. Here, I need to draw some empty space. This is called air sac. On the top of the egg, you can see a small empty space called air sac. That means air is present in this sac. That is called air sac. Okay, air sac. Now, in the bottom, we have some albumin deposit. So albumin deposit is present in the bottom of the egg. This is called albumin, 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 albumin. Okay, albumin present at the bottom of the egg. Then in the center, the most anticipated thing, the embryo is present. So embryo present at the center of the egg. So I'm just drawing a rough diagram of embryo. So this is the embryo. So embryo present at the center of the egg. So this is our embryo. Okay, this is embryo. Now embryo is always connected. Embryo is connected to the yolk sac. So here I will draw yolk sac. Embryo is connected to yolk sac. You will get that yellow color thing right in the egg. That is this one. This yolk sac will have all the nutrients that are required for the embryo. So this is the yolk sac. YOLK, yolk sac. Yolk sac. So this is yolk sac and this one is embryo. Now this embryo and yolk sac is submerged in amniotic fluid. This is called amniotic cavity. This full embryo is in the amniotic cavity. Okay. So even for humans, embryos are always suspended in amniotic cavity. So this is amniotic cavity. Amniotic cavity. AMNIO. Amniotic cavity. Amniotic cavity. Yeah. So these are the parts of the embryonated egg. So an embryonated egg has an outer shell. It has a double membrane, inner double membrane called chorioallantoic membrane. In between this membrane, we have an empty cavity or space called as allantoic cavity. In the center of the egg, you have the embryo. Embryo is connected to the yolk sac, and this embryo is suspended in the amniotic cavity of the egg. And on the top you have air sac, and in the bottom you have the albumin. Okay. Now, students, different viruses will grow in different locations of the egg. Say, for example, pox virus. Pox virus can grow in the cam membrane. In the pox virus can be inoculated in the cam membrane of the egg, whereas influenza viruses will be inoculated in the yolk sac of the egg. And some other viruses like coronavirus can be inoculated in the amniotic cavity of the egg. So there are few viruses that can be injected in multiple regions of the egg, and that is called inoculation of virus. Inoculation of virus. So how we will perform this inoculation means after 12 days of after incubation of egg for 12 days, first we will drill a hole on the top. We will drill a hole on the top of the egg. So first we need to identify the air sac of the egg, which will be on the top. Then we will drill a hole. Then with the help of syringe needle, we will inject the virus into multiple locations of this egg. Depending upon the type of virus we need to inoculate, we will inoculate the virus in multiple regions of the egg. For this purpose, we have certain techniques actually. First technique is called candling. Candling means first we need to know whether the embryo grown in the egg or not. How to identify the presence of embryo in an embryonated egg? That can be identified with a small trick. The trick is first you will take a lamp, you will take a light, okay, you will take a lamp and you will place the egg on a strong light like this. If you place the egg on a strong light, you can able to see some transparency. You can able to identify the embryo. So presence of embryo can be identified with this technique, and this technique is called candling. So egg will be placed on a strong light, in front of a strong light, and we will go for identification of embryo. If embryo is alive, if embryo is alive, embryo will start moving because of getting irritated by the light. Due to light presence, embryo will move. That indicates it's a live embryo. So we can go for this thing. And after candling, we will go for drilling. After drilling, we will inoculate the virus into the embryonated egg. Now let us see what viruses can be inoculated in what regions of the embryonated egg. So this is the embryonated egg. I said right. So we have air sac, we have chorioallantoic membrane, we have allantoic cavity, embryo, yolk sac, amniotic cavity, and albumin deposit at the bottom. So this is the structure of an embryonated egg, and these are the inoculation of various viruses in multiple regions of the embryonated egg. Say for example, in the yolk sac, we can inoculate herpes viruses. In the amniotic sac, we can inoculate influenza and mumps virus. And in the allantoic cavity, we can inoculate influenza virus and mumps virus and adenoviruses. And in the chorioallantoic membrane, pox viruses can be inoculated. So at least you need to remember one viral example for each membrane of the egg. At least you need to remember one or two viruses for each inoculation route of inoculation of viruses into the embryonated egg. So you can either inoculate into the chorial allantoic membrane, you can inoculate in allantoic cavity, yolk sac, or even amniotic cavity. And these are the viruses that we can inoculate in various locations of the embryonated egg. Okay, so in a nutshell, embryonated egg has four parts. We have chorioallantoic membrane, we have allantoic cavity, amniotic sac, and yolk sac. This is, say for example, this is a picture of a chorioallantoic membrane, students. So after inoculating chorioallantoic membrane with the pox virus, after 10 to after 20 days, okay, after inoculation of 10 days, I have harvested the egg. When I'm saying harvesting, that means I'm breaking the egg. I broke the egg and I took the chorioallantoic membrane. Now in this chorioallantoic membrane, you can see white spots, right? These white spots are actually called as POCKSS, poxes, POCKS, poxes, or nodules simply. Each nodule represents each viral replication. So one virus replicated here, one virus replicated here, one virus replicated here. So the virus has been replicated in multiple regions of this cab membrane. And this is how you can able to detect viral growth indirectly with the presence of detection of poxes in the surface of the chorioallantoic membrane of an embryonated egg. Okay, so this is all about embryonated egg and its things. So yeah, so usually, by the way, students, whatever vaccines you are taking now, you will get this one vaccine, uh, BCG vaccine, that is tuberculosis, and influenza vaccine. One vaccine you will get on your shoulder and you will have a permanent scar. Do you remember? Permanent scar on your shoulders after getting vaccinated in your childhood. Those scars are developed by a live attenuated vaccine, and all your vaccines are developed in an embryonated egg only. If you see any vaccine production industries, they will grow vaccines. That means they are growing the same virus as a vaccine candidate in the embryonated egg. So embryonated egg has a very big industry. All vaccine centers will use or heavily dependent on embryonated egg for the production of vaccines. Keep in mind. So embryonated eggs really plays a vital role in the vaccine technology. Vaccines will be developed or viruses will be cultivated extensively by using embryonated eggs. Okay, so you have a career opportunity to work even in um, vaccine centers, vaccine laboratories. So in vaccine laboratories also we have chances to get a job. So we can also work in vaccine centers. There we will handle these embryonated eggs. We will inoculate the specimens. We will ensure there is a proper quality, air supply, everything. So we have multiple opportunities for our career actually. So don't worry about your career. And also never stigmatize yourself or believe that you will only work in a hospital labs. No, not only hospitals, we have many other branches to work. We have vaccine centers, we have stem cell, blood banks, everything. Okay, that we will see later. So one interesting area is research such as vaccine centers. Okay, so yeah, this is all about the embryonated egg. Then there is a problem with embryonated egg. Not all viruses can be grown in an embryonated egg, and not all viruses can be grown in the animals also, as our Abhishek said. For example, HIV virus. I cannot grow HIV virus in a monkey. I cannot grow HIV virus in a rat. I cannot even cultivate HIV virus in an embryonated egg. Then how can I able to cultivate HIV? I don't have option of usage of animals. I don't have option of usage of embryonated egg. Then what is my third option? My third option will be I will use humans because only humans can have HIV, right? Will you agree? No, we cannot use humans for research purposes. Rather than we can take a piece of organ from the body and we need to um, grow the organ outside the body. We need to give proper nutrition requirements to the organ so it will grow, and we need to inoculate the viral. So that is our next chapter. So not all viruses can be grown in embryonated egg. So we need another technique, another approach for growing such viruses, and that is tissue culture. So viruses can be grown, or highly human specific viruses can be grown by the usage of tissue cultures. Now we have three types of tissue culture. The idea of tissue culture is first we will take a piece of tissue from the body. We will subject this tissue into some enzymatic reaction, so the cells will be uh, detached from the tissue. Then these cells will be grown in an artificial medium. This is called culture medium, tissue or cell culture medium. In the cell culture medium, we will grow the cells. Okay, these are cell line human cell line bottles. So now we got the human cells. Then we will go for viral studies on these cells. So our agenda is first we need to grow cells in a petri dish, and that is not a small task. It's a very challenging task to mimic living environment in a bottle or in a petri dish. They must get a proper oxygen, their metabolic waste must be removed, their carbon dioxide need to be removed, they need to get constant nutrients such as amino acids, glucose, everything they need to get. So it will take almost some 20 to 30 lakhs just to maintain a single cell line. Forgot about viruses first of all. Just to maintain a living cell line, it will cost around 20 to 30 lakhs. After maintaining living cells, now we need to sensitively we need to introduce virus and we need to wait for the virus to infect the cells and to see the viral replication. Very sensitive and very costly actually of usaging these cells. Okay, so this is the idea of using cells or tissues. So we have three types of tissue cultures. We have organ cultures, that means we will take an entire organ and we will cultivate it. We have explant cultures, we will take a bit of the organ and we will grow the organ in the petri dish. Then we have cell cultures. Cell cultures, we have three types of cell cultures: primary cell culture, secondary, and continuous cell lines. Now, I will tell you a problem with cell culture. 1 second, I will take another empty slide here. The problem with cell culture is let me reshare the PPT. Is everyone paying attention, students? Is everything clear so far? Am I too fast? Are you listening, students?
Yes, sir. Yes, sir.
Yes, sir.
Yes, sir.
Okay. Okay, thank you. Now listen, there is a problem with our human cells. I will tell the problem. Say for example, I got a cell. This is a cell, and I kept this cell in a petri dish. I kept this cell in a petri dish. This forgot about all the technical problems or challenges. Somehow we got a living a solution to give to sustain the growth of cell. So now cell can able to grow in this medium, but cells has a limit of their replication. All cells has a limit of dividing, and that is very important for us. See, a single cell can undergo replication for almost 10 to 20 times. 10 to 20 times. Okay, let us keep it simple, 10 times. That means this cell will replicate one time, given two cells. Okay, now this is first generation. Then these two cells will grow again. This is second generation cells. Like this, cells can go almost 10 generations. Within 10 generations, you will get enough quantity cells. So after 10 generation, 10th generation, what happens is cells will undergo something called apoptosis. And this is a normal mechanism of our cells. Apoptosis. What do you mean by apoptosis, students? Do you remember apoptosis?
The killing of cells.
Yeah, foregrammed cell death is called apoptosis. In the first semester, we discussed foregrammed cell death is called apoptosis. Now, that means by the time of getting 10th generation of cells, it will take almost, say, for example, 28 days. So by 28 days, I got 10th generation of cells. After 28 days, what will happen? All my cells will start senescing. That means all my cells will start dying in the petri dish. They will die within, after 28 days, there won't be any cells at all. So after 28 days, all my cells will be undergo apoptosis and there won't be any more cells. But can I use these kind of cells for replicating a virus which has 6 months incubation period? For hepatitis, it will take 6 months for the replication of virus, and my cells can only survive for 1 month. There is a huge block or hurdle on using cell lines. The problem is the cell normal death rate. It is limited either to 14 days or the highest will be 1 month. After 1 month, all cells will die. A very big problem. Cells can only replicate for a given period of time and a given number of cycles. After this, once they are reaching this number, they will die. After 10th generation, cells will go senescence. They will go apoptosis. So scientists become very confused. They want a solution for this problem. One day, an interesting story or incident happened. I will tell the incident. There is a woman. By the way, do you know what is cancer, students? Do you know any basics in cancer? What do you mean by cancer? Tell me, students. What is cancer? Can you tell me?
The unlimited growth of cells.
Very good. Unlimited growth of cells, right? Now, yeah, now the same idea came. I will tell. One day, what happened is, um, there is a family, a farmer family. They are tobacco farmers. Okay, they will cultivate tobacco plants, and there is a chance of getting cancers due to exposure to tobacco, right? So one lady admitted in a hospital. So her name is Henrietta Lacks. This patient Joy admitted in a hospital because of having severe stomach pain, severe pelvic pain. She has joined. Then doctor, there is a doctor. His name is Robert Gay. So this doctor observed the patient, and doctor recommended Pap smear for our patient. So they went for Pap smear. So they checked her cervical canal and vaginal canal. Now in the vaginal canal, doctor observed a very strange tumor. This tumor is in blue color. In this woman, she has a cancer actually, and her cancer tumor is in blue color. So doctor was curious, what is this tumor? And doctor is also aware of scientist problems like virology. He's also working on viruses. Then what the doctor did is, this doctor took a small sample of this tumor. Doctor took a small sample of this tumor and he went to the lab, and he suspended this tumor in a cell growing media. He suspended a, a cancer tumor taken from Henrietta. This Henrietta tumor he took and he suspended in a cell growing media. Then he started treating the patient. Unfortunately, our Henrietta died after 3 months of admission. So she died due to late stage cancer, and she is no more. But doctor was curious about her cells. He went to the lab. It's been almost 3 months. Okay, he observed the cells. Are they alive or not? They are still alive. They didn't underwent apoptosis. They didn't underwent apoptosis even after 3 months. Then doctor waited for 4 months. Still cells are not dying. He waited for 6 months. Something he started smiling. Still the cells are not dying. Okay, slowly he started telling this news to the scientists. It's been 8 months. Still cells are not dying. They're still dividing in the media. He sent samples to other labs. It's been almost 1 year. Still cells are replicating. Then they came to understood that Dr. Gay has discovered a deathless cells. They don't have death actually. They are immortal cell lines. Interestingly, students, this incident happened 60 years ago. 60 years ago, her name is Henrietta Lacks. Okay, Henrietta Lacks cells has been distributed into the world laboratories, and today we have almost 8 million tons, 8 million tons of Henrietta cells throughout the world, and those cells are still replicating. They are called immortal cell lines. And because of this accidental discovery, 17 Nobel Prize winners came. 17 Nobel Prizes came out of her cells. Today we are saying how many chromosomes present in our body, in our cell? How many number of chromosomes in a cell? Tell me, students. 46 chromosomes, or
21 pairs.
23 pairs.
20 pairs.
Okay, or 46 chromosomes.
Pairs.
How we came to know 46 chromosomes, number of chromosomes? Where we studied? We studied on HeLa cells, Henrietta Lacks cells. How we came to know about viral cancer genetics? Because of Henrietta Lacks. our recent Nobel Prize winner and Royal Society President, sir, his name is Ramakrishnan, Venkata Ramakrishnan. He studied ribosomes. He studied HeLa cells. We discovered HIV virus by growing virus in a HeLa cell. So there are 17 Nobel laureates who extensively worked with HeLa cells. And even today, HeLa cells are present in the lab. HeLa cells are so good at replicating that they become a lab contaminants actually. So if you are growing any cell media, that media will be contaminated by HeLa cells. So that much efficient these cells are. So these are called HeLa cells. HeLa cells means Henrietta Lacks cells. So they are called immortal cell lines or continuous cell lines. Because of this extraordinary discovery, the virology existed today. Because of these cells, we are studying viruses still today. Eventually, we got two more cell lines like pancreatic cell line, pancreatic cancer cell lines, all other. We got many cancer cell lines today, but out of them, very famous cell lines is Henrietta Lacks cell lines. So it really helped the scientific community. This story is really nice. But what is the contribution given to the Henrietta Lacks family? First of all, there is no informed consent from the patient to obtain her tumor. The contribution that has given to the Henrietta Lacks family is zero. They didn't get a single penny of credit from her mother's contribution from their families. Their children didn't get a single penny, even though everyone knows the fact. Eventually, with some fightings, with the help of one journalist, they claimed this problem to the court, and the court given some compensation to the Henrietta family. But it took almost, I think, 20 years to get justification to them. That's a personal problem. But yeah, this is all about in our virology. This is Henrietta Lacks' contribution. Okay, so we have organ cultures, explant cultures, and immortal cell lines. So the immortal cell lines are this Henrietta Lacks. Okay, HeLa cell lines. So this is our Henrietta actually. So she is Henrietta Lacks, and um, she got a vaginal tumor with a rare bluish hue vaginal tumor. So they took her, Dr. Gallo, Robert Gallo, took her cell lines, and eventually we came to know that these cells are immortal. They don't have any death, and we are using these cells for the replication of viruses. Okay, they are not dying actually. And interestingly, Henrietta cell lines uh, uh, have 30 characteristic mutations, and which is very rare. Because of presence of these 30 mutations, these cells become very good at replicating actually. Yeah, really interesting. So yeah, Henrietta Lacks died in 1951, and the tumor that killed her has been alive. That means still Henrietta Lacks is alive in one part actually, and growing to till this day. The tumor is immortal and was used to prognose polio vaccine, and is the jumping point for most human cell research to this day. Scientists have grown 20 tons of her cells. Oh, this is Henrietta Lacks cell. Uh, Let us see a short TED animation on Henrietta Lacks, then you will get some idea. Yeah, so just hear this one. Can you hear the audio?
Imagine something.
Can you hear the
Yes,
audio,
sir.
student?
Yes, sir.
Yeah.
Yes, sir.
Yes. Please continue. Yeah.
Imagine something small enough to float on a particle of dust that holds the keys to understanding cancer, virology, and genetics. Luckily for us, such a thing exists in the form of trillions upon trillions of human lab-grown cells called HeLa. Let's take a step back for a second. Scientists grow human cells in the lab to study how they function, understand how diseases develop, and test new treatments without endangering patients. To make sure that they can repeat these experiments over and over and compare the results with other scientists, they need huge populations of identical cells that can duplicate themselves faithfully for years. But until 1951, all human cell lines that researchers tried to grow had died after a few days. Then a Johns Hopkins scientist named George Guy received a sample of a strange-looking tumor. Dark purple, shiny, jelly-like. This sample was special. Some of its cells just kept dividing and dividing and dividing. When individual cells died, generations of copies took their place and thrived. The result was an endless source of identical cells that's still around today, the very first immortal human cell line. Guy labeled it HeLa after the patient with the unusual tumor, Henrietta Lacks. Born on a tobacco farm in Virginia, she lived in Baltimore with her husband and 5 children. She died of aggressive cervical cancer a few months after her tumor's cells were harvested, and she never knew about them. So what's so special about the cells from Henrietta Lacks that lets them survive when other cell lines die? The short answer is, we don't entirely know. Normal human cells have built-in control mechanisms. They can divide about 50 times before they self-destruct in a process called apoptosis. This prevents the propagation of genetic errors that creep in after repeated rounds of division. But cancer cells ignore these signals, dividing indefinitely and crowding out normal cells. Still, most cell lines eventually die off, especially outside the human body. Not HeLa, though, and that's the part we can't yet explain. Regardless, when Dr. Guy realized he had the first immortal line of human cells, he sent samples to labs all over the world. Soon the world's first cell production facility was churning out 6 trillion HeLa cells a week. And scientists put them to work, in an ethically problematic way, building careers and fortunes off of Henrietta's cells without her or her family's consent, or even knowledge until decades later. The polio epidemic was at its peak in the early 50s. HeLa cells, which easily took up and replicated the virus, allowed Jonas Salk to test his vaccine. They've been used to study diseases including measles, mumps, HIV, and Ebola. We know that human cells have 46 chromosomes because a scientist working with HeLa discovered a chemical that makes chromosomes visible. HeLa cells themselves actually have around 80 highly mutated chromosomes. HeLa cells were the first to be cloned. They've traveled to outer space. Telomerase, an enzyme that helps cancer cells evade destruction by repairing their DNA, was discovered first in HeLa cells. In an interesting turn of fate, thanks to HeLa, we know that cervical cancer can be caused by a virus called HPV, and now there's a vaccine. HeLa-fueled discoveries have filled thousands of scientific papers, and that number is probably even higher than anyone knows. HeLa cells are so resilient that they can travel on almost any surface. A lab worker's hand, a piece of dust, invading cultures of other cells and taking over like weeds. Countless cures, patents, and discoveries, all made thanks to Henrietta Lacks.
This is the story of Henrietta Lacks. So her, even though unfortunately she is no more, but she has contributed in the field of virology, especially on curing vaccines, providing vaccines to the world. So this is their actual family, and this is actually her granddaughter, Henrietta Lacks' granddaughter. She did the research. She been the vocal voice for getting the justice to their family. And interestingly, they made a series, a short serial or movie out of this Henrietta Lacks, and the serial is fantastic actually. So it was depicted by Oprah Winfrey, one of the best Academy Award winner. And yeah, you must see this one also.
For years, it seemed like a dream. About our mother, could this be true? What you don't understand is, we didn't know nothing about nothing.
Scientists had been trying to get cells to grow outside of the human body, but they would always die. Until Henrietta's cells came along.
I want to write a book about your mother.
Go HeLa, that's my mother. Hope I don't regret this.
Everyone's saying Henrietta Lacks donated them cells. She didn't donate, nothing. They took them and didn't ask.
The hospital is guilty, and somebody's going to pay. God wanna have a disease cure, he provides one for himself.
I don't know, Rebecca.
I'm not doing anything behind your back. Henrietta helped develop the AIDS cocktail, chemotherapy treatments.
It's hard not to get caught up in hope when you've been powerless for so long. I know I'm a part of you. And you a part of me.
It's very nice to see such documentaries once in a while. So we will appreciate our studies. We will appreciate everything what's going in these things. So that is what HeLa cell lines. So in this textbook, just seeing a word HeLa doesn't give much significance. But if you know the underlying story, if you know the background of these things, it will really give a sense of appreciation and a motivation to study further. So this is all about today's class, students. So in today's class, we discussed about viral cultivation. So viral cultivation can be done in three methods: by using animals, or by using tissues, or by using cell lines. So viral cultivation can be done by three types: animal inoculation, embryonated egg, and tissue culture. Embryonated egg, we can go with these things. There are certain advantages and disadvantages of those things. Then we got cell lines. We got in the cell lines we got three types: organ, explant, and cell cultures. In the cell cultures we got primary, secondary, and continuous cell lines. And the most famous continuous cell lines that we are using are immortal cell lines, which are HeLa cell lines, or the cell lines that has been derived from Henrietta Lacks. So this is all about today's class, students. If you have any doubts, you can ask me. Any doubts? I recommend you to watch that serial actually, Immortal Cells of Henrietta Lacks. That is the documentary. So thank you very much, students. I downloaded your attendance. You can leave.
Thank you.
Yes.
Thank you, sir.
Yes, students.